Right, welcome everybody, and welcome to another webinar by Technion-Australia. My name is Ori Danieli. I'm the Executive Director of Technion-Australia, which is the representative arm of Technion in Australia. And today, I always get very excited before we host those webinars because it's really a unique opportunity to meet firsthand with some of the movers of shakers of both Australia and Israel, and some of the most influential scientists of both countries. But there's one that I've been waiting for a while, and this is the one for two reasons. One, we're hosting three incredible scientists. One is Marcelle Machluf, who is I know she's a very modest woman, so I might get into a little bit of trouble of saying this, but she's nothing short of a rock star when it comes to science and innovation in Israel, and really been recognized as one of the most influential people in the country. So we're very privileged to have you here with us today. We've got Karlheinz Peter from Baker, who has got such a long CV, and I think we're not going to go into details today. I apologize to you, Karlheinz, because it will take probably close to the whole hour. And similarly, Thomas Marwick has got a very rich CV. He's the CEO of Baker Heart and Diabetes, and will host today's session and moderate the discussion. So just before I hand over to Tom to take over, I just want to go through some housekeeping rules. So as I'm sure everyone by now would be an expert on Zoom, there's a Q&A button at the bottom of the Zoom, and you're welcome to ask questions at any time. You don't have to wait for the end of the session. And Tom will do his best or Tom, Marcelle, and Karlheinz will do their best to answer. On that note, I just wanted to thank our partners today, which is Baker Heart and Diabetes Institute. And I'm very looking forward to hear more about what you have to say about innovation in this space and commercialization in the heart and diabetes sector. Handing over to you, Tom, and I hope everyone enjoys the session. Thank you.
Thanks, Ori. Thanks. That's a lovely introduction. And really, we're absolutely thrilled to be partnering with you in this session. I think it's going to be a very interesting one. My name's Tom Marwick. I'm the Director and CEO at the Baker Heart and Diabetes Institute. To those of you who are perhaps unfamiliar with the Baker, we're nearly 100 years old. We're an institute directed solely towards research in cardiovascular disease and diabetes. And a lot of what we do relates to commercialization and the development of new technologies and new drugs. So we're really thrilled to be discussing this today. So we have two presenters today. Both of them will talk about aspects of cardiovascular disease, starting off with myocardial infarction and then dealing with the consequences. So I'm going to turn first to Karlheinz. Karlheinz is a colleague of mine, a cardiologist also at the Alfred Hospital in Melbourne. He's an interventional cardiologist, and his interest is in dealing with vulnerable plaque. So Karlheinz, over to you. Tell us about what you're working on, and then we'll open up for discussion.
Thank you very much for the introduction, and it's a pleasure for me to speak on this topic. Just a moment. So my project really describes the development of a medical device that allows you to detect unstable plaques and essentially to prevent myocardial infarction. And the reason for this and for the medical need is really shown in the statistics on myocardial infarction. It's the largest single cause of death, so 1/5 of us will die on a myocardial infarction. When we have a myocardial infarction, 40% will die on the spot. So this is really the first manifestation, and you don't survive this. And the ones that make it to the hospital, only 1 in 5 really report in due time. In due time means that you still have a chance to salvage the myocardium that is not perfused and supported by a blood supply. So there is clearly a strong medical need for the prevention instead of treatment of myocardial infarction, because the treatment most often comes too late. Now I show you an example of a patient who I treated. So this patient presented to the emergency department with chest pain, and then we injected contrast into the right coronary artery, and you can see that the contrast doesn't really run down to the coronary artery. The artery is blocked here. And you see it better in the next picture. Here I push down a guide wire, and what you can see now is a thrombus sitting there and also an unstable plaque just above the thrombus that ruptured and caused the clot build up. And we have now quite fantastic tools available in the cath lab, so we can use balloons and stents and kind of reconstitute the flow, as is shown here. So you don't even see where the original plaque rupture and the thrombus was. And you can see this area is now quite well perfused. And if you make it in due time, so within 1 or 2 hours, we can salvage the myocardium. If you come too late, you have a large damage in your heart and you have problems to pump the blood, and often your patients develop heart failure. Now, most of us have actually plaques in their coronary arteries. And this is a study done a few years ago in Boston with patients, so people who died in car accidents, and the coroner looked at their coronary arteries. And for example, when you look at teenagers, 40% of them already have plaques sitting in their coronary arteries. And when you go up to the elderly population, probably 80 to 90% of the elderly people have plaques sitting in their coronary arteries. So in a way, most of us are lucky to have stable plaques, but some of us are not lucky. They have unstable plaques in their coronary arteries. And this is like a ticking bomb, because we don't know when it goes off or whether it goes off at all. So it's quite an unsatisfying situation, and we don't really have imaging tools available to find out which plaque is stable and which is unstable. Now, we developed a mouse model that creates an unstable plaque at a very predefined area. And you can see this here already. There is some bleeding in this plaque. It's a very unstable region with clots inside, a large necrotic core, thin fibrous cap. And when you compare it to a human plaque, it looks pretty much the same. And the big advantage with this model is now that we have stable plaques and unstable plaques in the same mouse. We use it to discover reagents that drive plaque instability and also to test drugs that stabilize plaques. And by chance, actually, a few years ago, we found, when we looked at these various plaque areas under a scanner, a near-infrared light scanner, that some of these areas suddenly developed a very strong autofluorescence. So this is the area of plaque instability. And when you shine light at 700, 800 nanometers on this, it has a strong autofluorescence. Initially, we were not sure whether this is a mouse phenomenon only, so we checked it in unstable plaques. In humans, we took carotid artery plaques from patients who had strokes, and they have exactly the same strong autofluorescence. And then we used our mouse model and just used a laser light scanner. And when you shine light into a mouse at 700 nanometers, then the whole unstable plaque lights up. And this is the area shown here, where stable plaques don't light up at all. So based on this, we submitted a patent application and launched a company which we call NearTech, for near-infrared technology. And what we would like to do is patients in the cath lab who are normally receiving an angiogram, and we are just looking for the narrow areas, we would now look for areas that are unstable as well, which can well be areas that are not stenosed. And we developed a guide wire that brings light into the coronary arteries, and we can collect the light that comes back through this autofluorescence, and then we know that this is an unstable plaque area. And this is the way we use this technology. So we insert this guide wire into the coronary artery, as shown here. So it comes up around the aortic arch. Then we go into the artery and shine it on the various plaques. And if we find this near-infrared autofluorescence, we know that we are supposed to stent that area and stabilize this plaque. And this way we prevent myocardial infarction. So we are now at the state that we got the patent granted in Japan and the US. we founded this company, as already mentioned, based on input from the Baker and Swinburne University, and we have recently appointed a very capable CEO, Matthew Hoskins, who runs this company. And currently we are in the final improvement steps on the catheter design. And the next step now is really to attract capital for the next development round. And we think that we probably need 5 to 7 million Australian dollars to develop this technology. Thank you very much for your attention.
Very interesting. Thank you very much, Karlheinz. It's a very nice presentation. So we're open for questions. While we're waiting for other people to send questions into us, I have a few thoughts myself. I think many of the people watching this may not really have a notion of the duration that it takes to develop a tool such as this. How long have you been working on this, and when do you expect to roll this out in sort of routine clinical practice?
Yeah. So the work really started over 10 years ago when we decided to develop this mouse model, because until then there was no mouse model available that reflected what we see in patients, these unstable plaques. And so that was really the beginning of the project. And then it was kind of a by chance finding that we saw this autofluorescence. And initially we thought we made a mistake, that we injected the wrong substance. And then we followed this up, we repeated it, looked at humans, and that was really the basis of the finding. And then we had this patent application and the initial publication now 2.5 years ago. And from then on, we started the development. So it's a long process, and it will certainly take another at least 3 to 4 years until we are, at a product that we can really bring into the clinic.
Yeah, it does take a long time. I love the part of this story that is based on serendipity. I think it's just such an important part of scientific training to be open to things that happen by accident. It ranges from all sorts of things, from, often junior researchers are anxious when their hypotheses are disproven, but actually that's really when the fun starts. It's when you start learning new things, right? And this good fortune that you had with this is similar to that.
When the PhD student came and reported me the results, he was really afraid of that I will blame him for doing wrong experiments. So it was an interesting experience.
That's very interesting. So we do have a question from the audience. Could the guide wire also break up clots that lead to strokes? So I guess in this is the idea that possibly with intervening there are also risks as well. What are your thoughts about that?
Yeah, so this guide wire is kind of developed for the coronary arteries, but we are considering a technology that we can use from the outside of the carotid arteries, and because there the signal is even stronger, and that's the alternative technology. But then this would be a non-invasive procedure, and we wouldn't be able to stent right away, as we can do this in the coronary arteries.
So there are two questions that actually fit in with that very nicely. One is, in the long term, is the diagnosis or detection part going to become non-invasive?
Yeah, so in the mouse, we can use this scanner technology. So this is similar to a CT. We just use light, and the mouse is not, is small enough to allow this. In humans, it's still, the sensitivity of the light detectors are still not good enough. So we don't have a CT comparable machine that would allow to do this light detection. At the moment, that will be available, and I think companies are working on that. That would be the non-invasive part.
Yeah, I think that is a really important aspect of this, because what we do need is a non-invasive tool, because after all, these plaques can change from being stable to unstable, and we would like to be able to repeat this. So another question here relates to the number of people that could be helped with this. How many, if you look upon this as kind of a population health intervention, how many lives could be saved by this process?
So we could use it in all patients who present with myocardial infarction and to get an acute procedure for the culprit lesion. So that caused the infarct. And then we would screen other coronary arteries and see whether these patients have additional unstable plaques. And we know that a lot of these patients, up to a quarter of them is the estimate, will have these unstable plaques. So the numbers are really huge when you know the thousands of patients or millions of patients really worldwide to have myocardial infarctions and are brought to the cath lab at the moment.
Yeah, I think this could be very impactful. I think more so with the non-invasive strategy, but absolutely along the lines of what you're talking about. I think potentially very important. Another question, how likely that stenting from this could cause in-stent restenosis? So again, that comes to the trade-off. You do something to reduce risk, but at the same time, the flip side of that is that you may do something that increases risk, paradoxically. What are your feelings about that?
Yeah, so we have seen massive improvements over the last year. So previously, when we just used bare metal, we had a restenosis rate of 20 to 30%, and that is now down to 1 to 2%. And what we do, we kind of immobilize drugs on the surface of these metal stents, and this prevents that we develop restenosis. And it's very rare, really, that we still see that. So this technology and progress really has solved this problem. That 20 years ago it was a major problem, but it's not anymore.
Yeah, I agree with that. I think it's this is one of those things that this is the time to do it, because had we tried to do this 10 or 20 years ago, I think this problem of restenosis really would have caused more trouble than we would be solving. Let's go back to the process of developing and commercializing this. We're cardiologists, we understand the biology here, but obviously there's a heap of other people that will be needed to develop a tool such as this. Who are you collaborating with, and what parts of this are out of your comfort zone?
Yeah, so we were quite lucky to find a partner here at Swinburne University. They have a Center for Optical Innovation, and there's one engineer, Paul Stoddard, who originally came from South Africa to Australia, and he's very experienced, and he drives the technological development of the whole device.
Yeah, and I think that there must be other aspects of developing this, just in terms of sort of the look and feel and the user interface of the device and so on, which we're used to using as clinicians, but actually there's a whole sort of specialized area of developing, right? Yeah,
I think it's the medical device industry is quite specialized, and through our CEO, who worked previously in companies in this field, so we have quite a lot of contacts, and that's what we have to use. I mean, it's really an interdisciplinary approach, bringing a lot of people and a lot of expertise together. And it's also an approach that potentially we have to go to the US to get more expertise. They have a much larger industry in this space, and a lot more of expertise than we have here in Australia.
I think that is a very pertinent issue, and that is, at what stage do these things, become international?
Yeah, I think the discussion at the latest will come up when we need to find funding for large interventional trials, because ultimately we have to do a trial that shows when we use this technology that we can actually save lives, so that when we define and identify an unstable plaque, then we would stent a patient, that this patient does better than a patient that is not treated this way. And this potentially, this clinical trial will need several thousands of patients, and it's really difficult to fund, and that goes in many millions.
That's great. look, I'm very aware of the time, and I think we should move on. Marcelle has been very patiently waiting there. You know,
here, so it's okay.
Of course,
I'm a good
you know, we're talking about a preventive strategy here, but we also know that many people progress on, have an infarct, and we need to do things that will try and preserve the tissue. So, Marcelle, I'm really interested in hearing your work in this area.
Thank you. I will share my screen first. 1 second. Okay, so first of all, thank you for having me on this interesting webinar, especially also the collaboration that I have with the Baker Institute. We just awarded a grant with Professor Julie Macmillan. So I'm really, really happy to start this venture. And I'm sure that it will lead, like I will show, to let's say amazing data, but also surprising one to my opinion. And as you mentioned, we are dealing more on dealing with the disease after it occurs, and especially the way of treatment. And in the case of myocardial infarction, we are all aware that we are missing therapies after myocardial is already diagnosed, and we need to help the patient, especially reducing inflammation and maybe preventing more deterioration of the cardiac tissue. So we at the Technion, I'm coming from the Technion in Israel, we developed a unique technology, which we already opened the company a year ago, and it is termed Nanoghost. And essentially, when we started to develop this technology, it came from ideas, different ideas in the lab, because my lab works on tissue engineering, including heart and trying to supplement the heart with tissue construct, but also with drug and gene delivery approaches. At the beginning, we started with synthetic material, trying to see how we can deliver the drug or to deliver the therapeutic to the site of interest, again, in the case of the heart, but not only, also in the area of oncology and cancer therapy. And that's where we started with this technology, but afterward I will show the switch, how we can use the same technology actually to target a different disease like the cardiac myofarction. So let's start with the technology Nanoghost. Before that, I will explain that all this delivery system or all this system is based on one unique cell, which is called mesenchymal stem cells. This type of cells reside in the bone marrow, and it resides in all of us, all human beings, also animal, and so forth. What is unique with this type of cell is that it can home to inflammatory site, for instance, like in the case of myocardial infarction, but also other inflammation. If related to today's problem like COVID-19, it is known that MSC, that these cells, in short, MSC, do deliver themselves to the lung to actually try and fight the inflammatory stage that happens after COVID-19 infection. So these are cells that are known to home to inflammation, but not only inflammation. They home also to tumor. And because a tumor also have a manifest of inflammation, it makes sense why they home to this type of tumor, to tumor in general. So in the case of tumor, what happens is that the tumor is calling the cells to protect itself from our immune system. So the cell come around the tumor and actually makes the tumor evade and not be identified by our immune system. Once the tumor grows, it leaves the cells away and tell them, okay, you can go now. And now it can grow by itself because the immune system cannot fight it anymore. It's too big for our immune to do anything to this type, to the size of the tumor that already occurred. So we asked ourselves, how come these cells, when they are after tumor or the inflammatory side, as I will show, they stick to the area? And apparently what happened is that these cells, when they come to the inflammatory site or the tumor site, they actually interact with the membrane of the cancer cells or the inflammation cells in the site. Once they do that, they work as they should work or not work, and then exert their efficacy. So the idea that we figured out that we don't want to use cells. We want to use something that we can actually control when we infuse it or inject it to the body. And in this case, we devised a system which is based again on these cells. Now we empty the content of the cell, we take everything out, and we leave only the membrane here, like we see in this diagram. And this membrane actually have all the recognition moieties that recognize the site of inflammation or the site of tumor. After we have that, we call this entity a ghost, because it's like a body, it's like a soul without a body or a body without a soul. You can choose whatever sentences are better for that. But the idea here that once we have this membrane, it cannot change according to physiological condition. Once we have this membrane, we downsize it to a small nanovesicle that can be injected freely to the bloodstream and can circulate all over the body. And this nanovesicle will stick to the site of inflammation or tumor, like the cells, only if we have a disease in our body. Once we don't have it, it will be cleared from the bloodstream. At this end point, we can add a drug, we can manipulate the vesicle itself to do some other therapies, but we have the technology to do all of these things. This is the movie that actually explains how we develop it for cancer, and then I will explain how we are going for cardio.
Chemotherapy is like trying to fight off a rat by burning down the entire house. The challenge here is to destroy malignant tissues without harming healthy ones. This is why targeted drug delivery is the holy grail of cancer research today. However, existing solutions are limited to certain kinds of cancer at particular stages. Huh? If only we could build universal carriers that can target multiple cancers at different stages while carrying diverse therapeutics and releasing them only upon penetrating the tumors. Doctors would finally be able to treat cancer much more effectively. Researchers from the Technion-Israel Institute of Technology have recently found a way to construct such carriers from the outer membranes of mesenchymal stem cells. Due to their natural role in tumor growth, which mostly involves protecting the tumor from the immune system, these cells possess a variety of surface-associated mechanisms, allowing them to target multiple tumors. The researchers, led by Professor Marcelle Machluf, developed a technique to empty these cells from their content, leaving only their outer membranes, then reducing them to nanoscale, creating nanovehicles called nanoghosts. These can be loaded and used to selectively deliver diverse therapeutics. Removal of the cell's content makes these nanoghosts inanimate and unaffected by the patient's body, allowing them to deliver a stable therapeutic effect over repeated administrations. Importantly, the nanoghosts retain their source cell's targeting capabilities, so they can travel in the patient's body until they hit a tumor, accumulate around it, penetrate in between and into the tumor cells, and release drugs to destroy the tumor. Moreover, by retaining the mesenchymal stem cell's ability to evade the immune system, such nanoghosts can be produced as an off-the-shelf product, requiring no cells from the patient himself.
So actually, this is the nanoghost, and now, okay, we developed it for tumor, but I mentioned that the cell themselves goes also to inflammatory diseases. So we wanted to study whether the homing capability of this nanoghost is not only to tumor, but also to inflammatory disease, like in the case of myocardial infarction. So we first tested this nanoghost on cardiac cells that were inflamed. And what we can see here in the image, in the middle, the nanoghosts themselves look like red dots, and the cells look green. And what we can see that once we place the nanoghost on the cardiac cell, they enter freely to this cell. So this arises the question whether we can now deliver a drug to this type of cells in the heart, of course, and then help them to evade or at least reduce the inflammatory stage of the tissue itself. This image, for instance, shows beautifully how the nanoghost in white interact with these cells and inside the cell and accumulate very fast. If we look at this graph, already in 24 hours, I can see the nanoghost filling all the cell. So we went to a model in vivo in which, like Professor Karlheinz explained, we initiated an infraction in the heart, meaning that all the area died because it had lack of oxygen supply, the vessel were clogged, and then we injected this nanoghost to the bloodstream. What we saw is that the nanoghost accumulated in the infract area, but did not accumulate in the normal or the healthy area of the heart. Well, to our opinion, this is a one-of-a-kind delivery system, because up to today, when a physician wants to treat cardiac myoinfarction inflammation or all the manifest around this disease, they give the patient drugs which are infused directly to the bloodstream. These drugs, some of it arrive to the heart, some of it not, and not always focus only in healing or helping the diseased area, but also affect normal tissue, and this is side effect that we are trying to avoid. Not only that, if we can deliver a certain, for instance, genetic material, like we have in our collaboration with Julie, is that we can change the regulation of inflammation inside the heart, and by that, reducing inflammatory process and maybe giving the heart the chance to heal itself. So we hope that this venture will end up with real data that, of course, will reach, hopefully, in the future to patient after clinical studies. And of course, I want again to thank the Becker Institute for granting us such amazing grant that will launch our venture into this area of myocardial infarction.
That's great. Thank you very much, Marcelle. There's really fascinating science there. Can I start off by asking you about how you take the original stem cell from the patient and then the time that is required to load it with the material that you do? How does that work at the moment?
At the moment, these cells can be purchased, first of all, because these cells are already approved for clinical application. Currently, there are different diseases that can be treated with the cell themselves. And because they are not immunogenic, they can be injected. And the problem was that they didn't show much efficacy like they should have. So company evolved around that and are producing these cells from biopsies taken from patients or from healthy donors. In our case, we take, we buy some of this amount of cells, we grow them in our facilities, and then we make the nanoghost. Once we have the cells, it takes like half a day, like around 6 to 8 hours to have the whole nanoghost in our hand. And loading, it's a few minutes or an hour or two to finish loading them with the drug and to be ready to be used for any studies.
Okay, that's a good answer. Marcelle, can I just ask you to turn off your screen sharing there so people can see us discussing easy?
Sorry.
No worries. Any questions from the audience? So tell us a little bit more about the timeframes that you anticipate here. So, you know, again, this is a story of laboratory discovery and then the process of translation. How long have you been going, and how long do you think it'll take?
Yeah. When I heard Professor Karlheinz talking about the time that it took him, and similar to what we heard, it took me 10 years. The patent was submitted around 2010, was approved 2 years ago, totally approved in the US, Europe, India, and now pending in China. But 10 years of research that accumulated to all the data that we had to have in order to convince the investors that we have a unique technology in our hand, and to launch the company that we just did a year ago.
Yeah, I think that this takes longer to build this scientific story than I think than people often realize.
You can't deliver technology if you don't have substantial data supporting not only its efficacy, also its safety. It's not if I have an idea and I tested several animals in a few months, then I can go and launch the technology and be used in human beings. And I'm happy that this is not the way that it should be done.
So how have you gone, you mentioned your investors. Tell us a little bit more, and perhaps that's something also for you, Karlheinz, about how they approach this from a market analysis standpoint. At what stage should that be considered?
Well, actually, we are considered as early stage because this is a startup company. All our data was proven in animal. In the field of cancer, I imagine like in the field of oncology, it's a big market, and therefore you don't have so many jumping on risk and putting their money when they think there is a risk in the technology, because it's not already a company. And it took me 3 years knocking on doors and trying to convince investor that we have a real thing in our hand. So in this case, we have a huge biotech VC here in Israel, actually established by high-tech people that do believe that the biotech is the future of our country, and decided after a whole year negotiation to invest initial funding in the company. And of course, this helped us to launch and to hire people and to start, and it will take us for sure other process in finding more money to go to clinical studies, because clinical studies, in our case, just the phase 1 clinical study cost like around $15 million to $20 million. So it will be another hard work to get this funding.
Yeah, no, I agree. So a couple of other questions coming through here. You may not be able to tell us this, or maybe if you do tell us, maybe you'll need to shoot us afterwards, but how do you remove the cytoplasm and then replace it?
Well, it's already written in the patent, so I can share with you. But
Good.
The know-how still stays in the lab and in the company. So what we do is we put the cell in a solution that actually cause the cell to inflate like a balloon. They become big balloon, but they don't explode. Once we have this big balloon, we have a method that is called homogenation. It's mixing. It's like shaking the cells. And once you shake the cells, because they are too inflated, you have like holes in the membrane, everything is dumped outside. So it's like you shake something until you get rid of, then you clean. There is a procedure, simple procedure of cleaning whatever went out, and still shaking again and shaking again until we make sure that everything is out. Once we have it, then we break it by putting energy. In this case, we're using a special mechanistic instrument which pressure everything through small holes. when you take people and you pressure them through a door, just imagine the huge pressure and what happens. So I don't want to imagine that on the Nanoghost, but it will break the membrane into small membrane that will close on themselves, and we have Nanoghost.
Yeah, okay. So a couple of questions related to that. There's a lot of interest in exosomes for in a similar way. Tell us about the pros and cons of the exosome compared to the Nanoghost for the kind of delivery stuff we're talking about.
Yes, this is our fitting market, we can say, although exosomes are not approved yet for clinical studies. So exosomes are very small entities, bodies, yes, similar to the Nanoghost. However, they are already produced inside the cell. And then you need to take them from the cell and clean them and use them, okay? And they have similar features to the membrane, but not all the feature, only similar feature. So now what happens is that you get small bodies, exosome, which you cannot change their size. The size that you have is the size that you need to choose. And therefore you are, let's say, limited with the capacity that you can load drug inside. They are smaller than the Nanoghost. Okay, so you are limited with whatever you can load inside. Not only that, you cannot empty the exosome. Most of the whatever they have inside stays there. And now they need to show that this do not cause any immunological reaction. On the other hand, the Nanoghost is a scale-up process. I can make the Nanoghost in any size of choice because I'm starting from a big cell, which have a 10 or to 50 micron and going to 200 nanometer, and I can range the size that I want. Not only that, because I'm making the Nanoghost from the membrane of the cell, I don't have the immunogenicity because I don't have anything but the membrane. Once I have some entities of the cell still in the vehicle, then I can also cause immunological reaction.
Very interesting. That immunological reaction makes me think about how the cell, how the ghost targets the cells of interest. Is this something that you've engineered as well? I mean, how have you thought about the process of targeting myocardium versus cancer cells, and do you intervene in that way?
Okay, so first of all, we are not changing the membrane. Okay, the idea behind the technology is that this membrane already have all the needed moiety. What also is very important, that is lacking the moieties that actually provoke the immune system. That's why these cells are called quiet cells and can be injected from one patient to another without any problem, and that's why we can buy them. So yes, they will target themselves to inflammatory process regardless of the where the inflammation occur. Okay, so a tumor is considered, for instance, a big inflammatory diseases. We know today that the Nanoghost, when they arrive to the tissue, for instance, even to the tumor, they interact with the immune cells inside the tumor. Similar, that's what they do in the heart. On top of that, if we want to target them better to the heart than to other organs, we can simply genetically engineer them before we make the Nanoghost. We take the cell, we genetically modify them, so then now they can express on their membrane a moiety or marker that recognize only cardiac cell. And that's what we did. So once we have this marker, then we have better targeting to the heart rather than targeting to other tissue or other diseases.
Yeah, very interesting. So that's a nice segue to come back to you, Karlheinz. I mean, if you think forward to when the kind of diagnostic work that you do happens non-invasively, potentially you may want to treat that medically, perhaps without an intervention. So presumably there are things within the vulnerable plaque that are potentially markers for vehicles such as this to deliver treatments. Have you thought about that?
So the stent treatment is one option, and as you mentioned, the medical treatment is the other option. And currently we have quite expensive drugs available with the PCSK9 inhibitors that kind of really reduce massively the cholesterol. So that would be a possible approach, that we identify the patients at risk, and then this would be a good justification for these expensive drugs. I think that's probably the medical direction, and we would hope that we will get more anti-inflammatory drugs. What we saw in these animal models is that plaque instability is very much associated with inflammation. And if we develop new antibodies against inflammation, I think this would be another class of drugs that potentially can be used in this setting.
Yeah, I think this story about inflammation is going to be extremely important to us in coming years. So a question here about the substances that are used in the nanoghosts. Marcelle, how stable is this? You mentioned that it's a product.
Yeah.
What's the shelf life of the product?
So we call it off-the-shelf product because we can prepare a lot of these nanoghosts and then store them. We can store them in -20 degrees or -80 without any problem. And we can store them for more than a year. For now, we tested them for more than a year without changing any of their, let's say, parameters or any parts of the nanoghost. And we also are shipping it to our collaborators just by using 4-degree shipping method. And they are pretty much stable.
So stable product.
Yes, it's stable.
So you've spoken a lot about how the nanoghost identifies the inflammatory cells, but I guess the next stage is actually delivering it into the tumor. So I have a question for you about how you approach that, and I'd also like to ask Karlheinz, because I know in his lab he has an approach using microbubbles for doing this. But to you, Marcelle, to begin with.
Yeah. Well, actually, again, I don't need to do much. The nanoghosts themselves do the work. What we found out is from 5 types of tumors that we already studied is that the nanoghosts fuse with the cell and actually dump their content into the cytoplasm of the cell. This is one mechanism. The other one is that they can dump their drug also outside the tumor cell. Because they are made from the mesenchymal stem cells, the tumor cells help them to, let's say, evade all the bulk of the tumor. We did so many sections in the tumor, we found them everywhere. Usually the delivery system, it comes to, let's say, to the surface of the tumor, then the drug is released, and we hope that the drug will reach as many as possible cells to treat them. In this case, because the tumor or inflammatory cell think that this is our MSC, they think, but this is not MSC, they help them to actually penetrate all the bulk and reach different area of the diseased area. So that's how they do it.
Very interesting. Karlheinz, tell us about your approach to this targeted intervention.
So we have kind of an alternative approach to Marcelle's very interesting targeting, because we found that when you have this ischemic myocardium, that they are, when you reopen the vessel, that the first cells that flood in are platelets, and platelets stick more or less to the previously ischemic myocardium, and we can use antibodies to target bubbles, for example, microbubbles there, or we can use drugs to target there. So it's essentially also addressing this very important problem that we have this so-called ischemia-reperfusion injury. When you open up the vessel, a lot of the immune cells flood in and kill actually the myocardial cells, and a lot of the heart failure that we see is caused by this immune reaction. I think Marcelle's approach and ours potentially will help this, and it's a very important area because we don't have any drugs available in the hospital at the moment.
Absolutely, yes, it absolutely is. So one thing that we haven't spoken about for the last 50 minutes is the word COVID, which is different from the rest of our lives, right, at the moment. Tell us about how this pandemic has influenced your work, Marcelle, and Karlheinz may be the same, and also about how you might use this kind of technology to deal with the pandemic.
In my case, we issued just yesterday a patent on the area of COVID-19. We have already developed what we call a decoy. Actually, as I explained with the Nanoghost, we engineered the MSC to express the receptor for the COVID-19 on the membrane. Then we made not Nanoghost, but Ghost. What happened now is that if you inject them to the bloodstream, they will serve as a decoy because actually they express whatever COVID-19 attached to. So if COVID attacked the lung, and we know that this ghost will go first to the lung, if COVID will attack to the lung, then it will attach to this decoy and actually be cleaned from the lung and not be available to attack normal cell. This is one approach of the technology. The other approach that we developed is a vaccine, to our opinion, a safe vaccine that we are testing now. We are not a pharmaceutical company, so we are not doing it so fast. But again, going to the Nanoghost technology, here in this case, we engineered the cell to express the spike, the corona protein that we see on this virus. And once we expressed on the surface, we made nanoghosts, which are similar in size, like the virus. The virus is around 120 nanometer, and the nanoghosts are 150. Now, the nanoghosts express similar corona on their surface, and these days we are testing it in animals to see if it evokes the immune system to produce antibody against. The safe approach is that we are not using a virus. So this entity cannot cause damage if by any chance they will arrive to the wrong tissue, or if the virus will came alive if we're using dead virus or something like that. So any part of the virus is not there, only the corona itself.
Very interesting. And Karlheinz, for us as cardiologists, we know that one of the effects of COVID is on the heart. What are your thoughts about the story with the microvasculature, and does it have an effect on plaque stability, for example, and how about the inflammatory effects of COVID?
Yeah, so it turned out that microthrombosis is a major driver of COVID mortality, and so platelets seem to be very much involved in adhering on the endothelial cells that are infected by the virus. And you have these kind of microthrombosis in the lung, in the heart, in the kidney. So it's one of the major reasons why patients really die on COVID. And so the importance of having anti-platelet drugs, having anticoagulants given early on in patients has become very clear over the recent months. And that's an area, because we work a bit in developing anti-platelet drugs. And that's an area where we think our drug developments would be very beneficial. In regards to myocardial infarction, it's kind of a paradox situation that a lot of patients stay at home, so they don't dare to go to the hospital because they are afraid that they get infected. And so I think the mortality at the end with myocardial infarction will be higher when the data come out. And we saw a reduction of the patients coming to the hospital, and also the ones that come are often coming too late, and so the mortality of these patients seems to be higher.
Yeah, I agree. I think that certainly in places that have not had huge numbers in the pandemic, the other unforeseen consequences, such as what you're describing, may very well be a cause of more mortality than the virus itself, unfortunately. Yeah, absolutely. Yeah. Well, I think it's been a really fascinating discussion. I think it emphasizes quite a number of things. I think, first of all, about the importance of the inflammatory response at both stages, really, in the disease of plaque rupture and also in the consequence of myocardial infarction. And I think the other thing that it shows is really how long and how many experts it takes to develop a new technology like this, and also the pathway of commercializing something from discovery at the lab bench to a product is a long one and quite a challenging one in terms of developing it commercially. I'd like to congratulate both of you for the work that you're doing in this space. I think it's been a fascinating evening, and I hope we have an opportunity to have a similar discussion in the future. We at the Baker have a long-standing association with Israel. In particular, several of our groups have a collaboration with the Technion, and we love your science, and we also love the setup that you have for commercialization there, which really is a model for us to follow in Australia for taking our discoveries to actually a product. So Ori, thanks for giving us the opportunity to share the evening with you. It's been really fascinating, and back to you.
Thank you, Tom. Thank you, Marcelle and Karlheinz. It was very, very interesting, and I'm sure we could have gone for much longer, but we try to stick to the time here. And I want to take this opportunity to invite you to a few upcoming webinars that are coming up. We've got a very busy November ahead of us. Our first one is on the 10th of November with Professor Alon Wolf, which Marcelle knows very well. He's the vice president of the university, and he's quite remarkable in his own right. He invented a robot snake, essentially, that can crawl into disaster zones after earthquakes or stuff like that to find survivors, and later actually adapted the same technology to develop a snake, a much smaller snake that can actually crawl inside our bodies and identify different things. So he is going to speak alongside a UNSW alumni about medtech and the future of medtech, especially in a COVID world. Well, a week after that, Tom just mentioned Israel's capabilities in commercialization. So we're fortunate to host a partner at Jerusalem Venture Partners, which is one of Israel's most successful venture capital firms, with about $1.4 billion in management. And she's gonna speak about Israel's unique approach to entrepreneurship, innovation, and startups. And that's an event we're doing alongside with the Australian National University. And then later in the month, we're hosting Ido Kaminer from Israel, who is is a young professor, is very up and coming and an expert in quantum computing and quantum mechanics. And he'll be speaking alongside one of Sydney Uni's famous scientists, Ben Eagleton, about that topic. So you don't have to remember all that. I'm going to send everyone an email with links. We look forward to hosting it. Last point before we sign off, once we sign off from this webinar, you'll have a short survey. It will literally take 30 to 40 seconds to complete. It will really help us to kind of direct our content and make sure that we're on the right page. And we appreciate you all coming. This event will be recorded. You can visit our YouTube page to see this and all our 12 other webinars that we've done so far, as well as on our Facebook page. Lastly, thank you again to Baker Heart and Diabetes Institute for partnering with us and for supporting our teams back in Israel, Marcelle in this case, and of course to our speakers. We look forward to seeing you all again soon. Good night and take care, or good morning if you're in Israel.
Night. Bye.
Bye. Thank you.
Thank you very much.